Thank you, and thank you very much to the organizers for inviting me to speak. So, as it says here, I'm going to talk about our Huntington's disease program, but in the context of some of the chemistry advancements that we've made, but also touch a bit on some of the practical aspects of how we have decided to pursue this particular disease. I probably don't have to go through the history of oligonucleotide development here, but it's a long and very interesting history that began almost in the 70s with the realization that to improve the properties of an oligonucleotide, you can substitute a phosphodiester bond with phosphorothioate linkage, and that really changed the whole approach to oligonucleotides, where you could stabilize these molecules and potentially use them as a therapeutic. It was known at that time, however, when you do that, you create a chiral center around that phosphorothioate linkage, and you do create mixtures of stereoisomers as part of that process. But there were advancements over the years throughout that where there were chemical properties of these molecules were able to be changed, to add 2' modifications, to do other things, to continually improve upon this technology. And the success of this is it did lead to approved products over the years, and recently even more products for really difficult to treat diseases. Now, WAVE came on the scene a few years ago, where it was, as I said, it was known that these diastereomeric mixtures of oligonucleotides through the introduction of these phosphorothioate linkages was an issue which made it a little more difficult to control the pharmacology of these products, but WAVE developed a technology that allowed us to control the phosphorothioate linkages, the stereochemistry around those linkages, and potentially apply a more small molecule approach to the development of oligonucleotides. And that's essentially what you're seeing here, is when you introduce that phosphorothioate linkage, which is putting a sulfur in the non-binding oxygen, you create these chiral centers and you create these molecules that are essentially diastereomeric mixtures. WAVE's approach is to be able to control that and create what we call stereopure oligonucleotides that allows us to optimize the profile. And what that does also is it allows us to, over the years, we've developed this in a way where now it becomes, allows us to apply versatility of what has been learned over the years about oligonucleotide chemistry to these stereopure compounds, where through the creation of RP, or rightward-facing, or SP, leftward-facing monomers, it allows us to do more rapid candidate screening, to the point now where 96-well plates we're able to optimize and select candidates. It also allows us to scale. Back when this was first being thought about, it was, as I said, it was a known limitation of the development of oligonucleotides. Part of the challenge was the inability to scale up to quantities that are useful as a therapeutic. And through these innovations in our chemical, essentially our raw materials, we're now able to create a high purity crude molecule that allows us to optimize for a single compound. So the applicability of this is quite broad. This is a snapshot of our pipeline. We currently have studies ongoing in Duchenne muscular dystrophy, as well as Huntington's disease. We're actually moving towards studies in ALS, FTD, and we have some research programs in spinocerebellar ataxia and other CNS diseases. There's applicability in retinal diseases, and we have a partnership right now in liver diseases with Pfizer. But today we're going to be focusing on the Huntington's disease programs. Just a few words about Huntington's disease. So Huntington is a autosomal dominant disease characterized by cognitive decline, psychiatric illness, chorea, eventually leading to death. There are no approved disease-modifying therapies, though there's been great strides in some of the different approaches to this disease, as you may have heard throughout the meeting. The expanded CAG repeat of mutant Huntingtin, or the gene HTT, leads to the production of a toxic protein that begins to accumulate in the brain, leading to neuronal death. Now, normal Huntington, wild-type Huntington, is actually an essential protein for neuronal health and neuronal development. So you have the good protein from one allele, the bad protein from the expanded allele, and it's this balance of the two that eventually leads to progression of the disease and the fatal, detrimental long-term consequences of the disease. And although it's considered a rare disease, it's not that rare. We've got about 30,000 at least patients in the U.S. But that's really the smallest part of the population. This is an autosomal dominant disease. So there are a lot of people who carry this who will eventually develop the disease who are currently asymptomatic. So one of the great hopes of developing a therapeutic is that you can treat these people because it's a 100% penetrant disease. You may be able to intervene and change the overall course of the disease. So our approach to treating Huntington's disease is through allele-specific silencing. There are various approaches that have been explored where the overall Huntington protein itself, both wild type and mutant, are knocked down. We've chosen to take the approach where we're trying to just target the mutant transcript, hence the mutant form of the protein. And we're doing this through the association of certain SNPs that have been described in association with the long CAG. So there are certain SNPs that actually sit on the long CAG that allows you to specifically target the mutant allele to hopefully bring down the toxic protein. So we're utilizing the advantages of our Stereopure chemistry to increase the selectivity and potency of the ASO to hopefully give us that ability to differentiate between the two transcripts. So the SNPs that we are targeting are 362.307 and 362.331. I'll call them SNP1 and SNP2. And these are known SNPs that are associated with the CAG expansion in about 2/3 of the Huntington's population based upon genetic studies in the North American population. So we thought that by targeting these two SNPs, either one of these SNPs or both, we would be able to treat a large portion of the Huntington's disease population in a very specific way. So we developed two oligonucleotides, one that targets each one of these SNPs. Now, the next few slides, I'm just going to walk you through how we do our screening process. And this is, in general, especially when we're targeting a SNP, this is an example from Huntington's disease, but it can really apply to any other SNP targeting approach. So one of the first things we do is we have two RNA wings with a common DNA core, and we'll walk the SNP sequence through that DNA core, fixing it for the blue, meaning SP, so leftward facing bonds on the DNA backbone. And we create a whole series of these oligonucleotides, again, walking the SNP through, and then we put it in an assay where we're looking at iPSC-derived neurons that are cultured over a week, free uptake of oligonucleotide, because one of our primary screenings is whether the oligonucleotides go in without a transfection agent. It's one of the things we optimize for. And then we grow them for a week, and we see, does the oligonucleotide target, in this case, target Huntington, and knock it down. So what you're seeing is the bars represent basically fold relative to the negative control, which you see there as the brown bar. Now, what's shown here is that in this particular case, through this walk, we actually don't really get, you have three types of chemistry, and we really don't get very much knockdown at first, but across this whole oligonucleotide panel using this approach. Now, when we see this, that we don't stop here because, as I said, there's a lot you can do with stereochemistry through this to see if we can optimize further. So what we'll do is we'll pick one of those RNA wings, and then we'll fix the SNP now, where we want to be targeting. And then we do an RP walk, in this case an RP walk. So you have basically that SP core with an RP, as you see in red, that then just basically walks through the core looking for, does the walk of that RP, exact same sequence, exact same wing, change any sort of the properties of the molecule in terms of its binding. Same type of culture in iPSC neurons and derived neurons. And what you're seeing here, just focusing on these three molecules, these three, this is the magic here of the pointer. Well, you'll see that in the box you have those three oligonucleotides on the left. They correspond to oligonucleotides 24, 25, and 26 on the right. So what this illustrates is that depending on where the RP is, you get very different target engagement and knockdown of the mutant transcript. But not only does this show that, but if you do focus on 24, 25, 26, just by moving that RP linkage in one position 3', one position 5', you actually get very different potencies of the molecule. So that RP positioning does have an effect on oligonucleotide activity, and we then incorporate that into our optimization process. So now we say, okay, so we have an area where we are able to knock down, we have a SNP in a position we want it, we have an RP in a position we want it, let's optimize further. And the next step is, can we figure out which one of these now help differentiate between the wild type transcript and the mutant transcript. So this is this is a luciferase reporter assay where we're, again, varying the RP position now, but this time we're looking to see, can we differentiate between a SNP on one allele or the other. And what you're seeing here is this oligonucleotide 24. You get very nice differentiation in this assay between the targeting of the wild type transcript and the mutant transcript. So this would then be the molecule we then carry forward for further optimization. And then what we go is we then go back and we redo the wings. And we look at the other 2' chemistry. And you can see here that when you take oligonucleotide 24 and you change that wing chemistry, all right, at this time now leaving the RP exactly where we had it before, just by changing the wing chemistry and applying other 2' modifications, you get a different, let's go back. You get different potencies in terms of just by changing now, taking that original that RP targeting or that SNP targeting molecule, changing the wings, you can have great real differences in potency. As you can see, oligonucleotide 24 is the one we would be taking forward. So that sort of walks you through how we would get to a SNP targeting compound, and also sort of shows you that these subtle changes in stereochemistry, which really is just the changing of the stereochemical orientation of a single bond, can have large differences in potency and selectivity. So now that we've developed these compounds, we'd have to figure out how we get them to the clinic. And one of the most important things we came across was we have to be able to find these patients. We have to be able to detect the SNP in the population in a quick enough way that allows us to pre-screen these people to get them into a study. Because now we have 2 SNPs we're interested in, 2/3 of the Huntington's population. We have to not only detect is the SNP there in these people, but is it associated with the long CAG? Because you don't want to knock down your wild type protein. You want to knock down your toxic protein. So it has to be in association with that long CAG. So we developed an assay for that purpose. And this is just a flow chart that goes through the assay that we developed. We'd first have patients come in for pre-screening. We collect blood and we look to confirm that they have CAG number greater than 35, which would indicate that they are confirmed as having Huntington's disease. Then with those samples, we will look using PCR and Sanger sequencing to confirm that the SNP of interest is both present and that it's heterozygous. So again, we don't want a homozygous in the study. We want the SNP to be associated with that long CAG. Once we're confirmed that they have the long CAG and that the SNP is heterozygous for the SNP, we then use long read sequencing. And in this assay, we use the PacBio RS2 long read sequencing to confirm that the SNP is now phased with that long CAG on that one allele. And that's basically the process that then, if we've confirmed now that the SNP of interest is on the allele with the long CAG, that then allows us to put those people into the screening process for one of the 2 trials. So a SNP1, someone with SNP1 or the 307 SNP would go into our SNP1 targeting study. Someone with SNP2 or the 331 SNP would go into our SNP2 targeting study. And if people have both, which some people do have both, they would either go into one or the other. They're eligible for either because it knocks down the transcript regardless if you have 1, 2, or both. It just depends upon where we are with our recruitment. That whole process, when we started, took about 4 to 6 weeks. Now we're down to under a month where this can be done, and we have 2 commercial labs that can do it as part of our trial. And so when we developed this, we first started by looking at it in collaboration with the CHDI Foundation, which is Huntington's research foundation, where they actually had samples that they knew the phasing. So these are HD patient-derived cell lines that they actually knew had the SNP and knew that it was phased with the long CAG. So in a blinded fashion, they gave us these samples, and we went through the process that I've just described. This is the results of that assay, where you have the 12 samples on the left-hand side, and is your SNP1, SNP2, and then how good we were at detecting basically the SNPs that that CHDI already knew about. You can see that there were people who had both SNPs. You can see that there were people that had SNP1, some that had SNP2, some that had neither. So the blank ones had neither. So in this way, we had 100% concordance in this blinded assay to determine that our phasing process and the probabilities that we were assessing and the algorithms that allowed us to do this were robust. We then expanded it into a 200-patient research study, an observational study, where we looked in the U.S. in a practical way, can we take a large number of Huntington's disease patients, put them through this process, and can we successfully make a determination of their SNP? And second of all, nobody had done this in a prospective way before. A lot of the work on this had been retrospective analyses about the frequency of these SNPs in the population. So we wanted to determine, was the literature correct that about 2/3 of patients had these SNPs, and could we do it in a large-scale enough way to be able to conduct a clinical trial? And this also pre-identified patients should we then advance the SNP targeting therapies that we knew had these SNPs, and they could potentially enter a trial. So this shows the results of that research study. Again, on the right-hand side, you'll see sort of the flow chart that I've already described a couple of times, where we had people come in. Did they have the long CAG greater than or equal to 36? All of them did. You can then go that about 2/3 or about 3/4 of those patients were heterozygous for the SNP. The homozygous patients, about 1/4 of them. Then we had a few patients that we were unable to phase, but then of the ones we were able to phase, you can see that the vast majority of them had the SNP in association with the long CAG. And on the left-hand side, you can see overall, that gives us a frequency of SNP1, SNP2, or both of about 60%. So very consistent with what was known in the literature, and reassuring to us that as we brought forward these therapies, we were able not only to conduct trials to assess them, but also have the ability to have a commercialized product potentially someday and target a large part of the population. So now, just moving on to the actual clinical trial. So we do have 2 clinical trials ongoing now, phase 1b/2a studies, where with ASOs targeting SNP1 or SNP2, and I've already shown you the ability of these to knock down and differentiate between wild type and mutant. These are running simultaneously. They're global clinical programs. They're multi-center. They're double-blind, randomized, placebo-controlled. The drugs are being administered intrathecally in a single ascending portion of the study and a multi-ascending portion of the study. The primary objective of the study is to look for safety and tolerability of the intrathecal doses. These are what are called early manifest HD patients, so they've been recently diagnosed. They're considered stage 1 or stage 2 Huntington's disease, so relatively early in the disease. One of the key objectives is to measure the total and mutant Huntington protein in their CSF to essentially establish this proof of principle that we're able to not only just knock down Huntington protein, but make a differentiation between the mutant and the wild type form. The key inclusion criteria is listed there, and right now we're expecting to enroll about 50 patients per trial to accomplish this. In addition, there's an open label study, extension study plan to allow continued observation to better optimize the dosing frequency. They're getting it monthly in the current trial, but to understand whether we have persistent knockdown for extended periods of time. And one of the key things we want to do is not only expand this in the early manifest, but also, as I mentioned earlier, the pre-manifest population treating before people develop disease, which is the ideal in many of these genetically defined neurological diseases, or any neurological disease for that matter. But certainly here, where you can pre-identify these people, hopefully be able to get that pre-manifest population. And if we can show differentiation between wild type and mutant, that's going to make it easier to enter that population because they're younger in their disease course, even children, where the knockdown of wild type may have even a greater impact. This shows the study design. This is a SAD multi-ascending dose study. So the way this is designed is patients get, and it's done consistent with the European guidelines on first-in-human studies, where you basically, patients come in, they get a single dose, one patient with active, one with placebo, observed for 48 hours to make sure there's no SAEs, and then dosing continues up to a total of 12 in a given cohort. 4-to-1 randomization, just to, but with placebo. And then there's a dose escalation committee review in addition to a safety monitoring committee review where they're looking at all adverse events, any procedural related events, whatever is necessary. And if they're comfortable with that, the patients then go into a multi-dose phase and you begin the dose escalation on the single dose phase. So you have basically the single dose escalation happening as patients go into the multi-dose, and then the multi-dose data are looked at that allows the multi-dose dose escalation to occur. And it's a stepwise fashion. It looks like it's going down, but it should be going up. The doses are going up over time. And then eventually you have a wide range of doses in approximately 50 people. And the primary endpoint, as I said, is looking at the safety, but also looking at the production of the protein. This just reiterates what I said, the safety of the single ascending multi-dose dosing, looking for the protein, looking for PK. We're also doing imaging as well as other functional outcome measures. It's a really short study for determining functional outcome measures. These are very insensitive measures, so probably not enough duration or size to be able to assess them, but we're looking at them nonetheless, and we're looking at a variety of other biomarker outcomes, such as neurofilament light chain and others. So to wrap things up, HD is a pretty terrible disease. It's really one of these diseases where in an autosomal dominant fashion you can have a monogenic disease that allows us to really target what we believe is the core toxic protein in the disease itself, and is invariably fatal in the setting of that expansion. We have these SNPs that should allow us to differentiate between the good form of the protein and the bad, and hopefully allow us to then reduce the mutant protein and improve functional outcomes. The studies are ongoing, and over by the end of this year, we should have results from those studies, both in terms of the degree of knockdown as well as whether we can differentiate between the wild type and mutant. And just looking to the future, you know, as I mentioned, this is sort of was our first entry point into using stereopure molecules in neurological diseases. We have the studies ongoing in DMD, and we have a variety of other CNS targeting compounds that we're continuing to evaluate, where we're looking at can we do things, and this is MALAT1, which is a reporter gene. We're looking whether we can, using different chemistries, increase our potency by, again, applying stereochemistry, but as well as other innovative chemical modifications to the backbone, as well as both in vitro, in vivo, and importantly, in this disease, and you can see all the way over in all diseases, if you're going to have a CNS disease that you have to treat intrathecally, the goal is to make these therapies as durable as possible. And we're learning more and more about what are the elements of chemistry, in addition, in that interplay of sequence chemistry and stereochemistry, allow us to increasingly optimize how long these molecules can last, so that we can reduce that frequency of, because monthly intrathecal forever is not exactly an ideal scenario. But as we develop additional chemistries, we're finding there are key chemical modifications that influence that, and even key modifications that allow us to better access the nucleus to increase the potency and those types of things, even without transfection agents. We're learning more and more about how important stereochemistry is to basically all aspects of pharmacology. So with that, these are just the sites who did all the studies, and thank you to the team and the patients. A lot of patients and their families were part of these studies, and couldn't do it without them, certainly. So thank you for your time.
Hi there. Great talk. So another disease with trinucleotide repeat expansion is myotonic dystrophy, where the toxicity is coming from the RNA.
Yeah.
I was wondering if you have tried to look at that.
Yeah, we thought a lot about myotonic dystrophy. I think one of the things that we're working on there, as well as others are working on, is based on, there have been great studies in myotonic dystrophy from Ionis, and I think that one of the challenges there is access to the actual target in the muscle, right? In addition to, as you point out, the targeting of the RNA. So I think that it's something we're interested in. We're working on technologies to better access the muscle, and that might open the possibilities of going in that direction.
So you feel that the bottleneck over there is the delivery?
There's the delivery aspect that has to be worked on. With our DMD program, we're learning a lot about the things that influence how well you can get into muscle cells and how well you can access the basically myoblasts and mature muscle. And in myotonic dystrophy, obviously, the more we can optimize that, the better we'll be in a better able to approach myotonic. We're focused on DMD first, where you have muscle that's leaky. Makes it a little easier than in the setting of an intact muscle.
I brought the question because Ionis is using antisense oligon here to speaking about.
Well, that's what we're using too, yeah. So we're using antisense oligo, but we also are able to apply the stereochemistry to other modalities, and those are the types of things we're working on. But we're learning from our antisense oligo program in Duchenne that'll help advise whether we're able to do myotonic. We haven't really made any decisions about that. Yeah, thank you.
Yeah, the PacBio long-read phasing seems really interesting, and I'm wondering whether you can use that information. Have you identified kind of other SNPs, apart from 1 and 2, that could help identify some of the other individuals that may be either homozygous for some of those, or
yeah, we actually are, part of the work we're doing now is we have a third SNP that we're optimizing to carry forward that we've identified. Obviously, the longer it is away from the CAG, the more challenging it is to actually develop the diagnostic. The PacBio RS2 is the first generation of this. We've already now have different platforms, like an Illumina-based platform that allows us to maybe do it even faster. So we're continuing to evolve the assays that enable us to do this in a better way. Because the RS2, I mean, we're looking at the next generation after the RS2, both from PacBio as well as the Illumina platform, to try and get this going even better. And yes, there are other SNPs. There are 3 other SNPs that we've seen that are of interest.
Maybe just a quick follow-up. Is there anything unique about the CAG itself, now that you've been able to phase that entire length? Is there any internal variation?
There's a lot of, there's a lot of that. We haven't done that, but there is a lot of literature out there on the somatic expansions of the CAGs in the brain tissues and stuff like that. We haven't done that, though. We're just really focusing on making sure that SNP is in association with the long CAG. Sure.
Question to that one, in fact. So we know that the population's substructure of SNPs is quite different in different populations. So you showed a nice correlation in the North American population in your observational study. Are you also planning to look at this in other populations?
So, right now we've looked at SNP associations for long CAG and other diseases, like looking at some of the other things we're looking at, like the ataxias and other. So we are looking for those types of associations. But right now, what we're focusing on is just these particular SNPs and the ones we've identified for the next batch of studies that allows us to get a larger number of HD patients.
For the clinical compound, what's the beam chemistry?
So we haven't really disclosed the chemistry at this point, that'll all. I mean, from the first series of chemistries, we've evolved the chemistries. So depending on the compound, there's different chemistries, but that'll all eventually become public. We just haven't disclosed that.
All right, thank you. I think we need to move on. Thank you very much.
Thank you.